Preparation Model Paper 13. I'm Vinay Shankar Reddy, PhD Plant Pathology. In continuation to this, fill in the blanks that was started with the previous video. So this is just continuation. So most of the plant pathogenic viruses are transmitted by. So majority of the plant pathogenic viruses are generally transmitted by two kinds of vector, aphids and whiteflies. So if you see this, the aphids and whiteflies alone transmit around 60% of the plant viral diseases. So the first importance goes to aphids. So aphids around 50 to 55%. 45 to 50%. The remaining is white flies. These alone contributes around 60 to 70% of the entire viral pathogen transmissions. So the answer is aphids. The one or two club-shaped fruiting body is the characteristics of a division or a subdivision they are asking. So division, you can take division also. For example, production of asci is the characteristic feature of ascomycota, whereas the production of club-shaped basidia, if you see the basidia is club-shaped. So on the basidia, basidiospores are produced. Each basidium contains 4 basidiospores. Each ascus contains 8 ascospores. What they are asking is the club-shaped fungi. If you see this, basidiomycota is also known as club-shaped fungi, right? So they asked a subdivision. That's why it is a basidiomycotina, because the subdivision always ends with mycotina, right? The 103. The toxin produced by the Trichoderma viride. Trichoderma viride generally produces various kinds of toxins. For example, viridin, viridicatin, such kind of toxins will be there. But the most important toxin is viridin. Viridin is the most important toxin. And the 104, seed produced in a bacteria during spores produced in a bacteria during unfavorable conditions. For example, if you see fungi, in fungi the spores produced during unfavorable environmental conditions are like Sclerotia and Chlamydospores. We can say the fungi can produce during unfavorable time. For example, if it is a sexual spores, we can say oospores and zygospores. Those are the sexual spores which are produced during unfavorable time. For long-term survival during unfavorable time, this oospore and zygospore can survive. Whenever the favorable time occurs, again they start to germinate and the cycle will continue. Here the spores are produced by bacteria during unfavorable time. Generally the bacteria produce no spores except one bacteria, that is Streptomyces, right? So normally the bacterial spores are known as endospores. Bacterial spores are known as endospores. So the answer is endospore. Coltotrichum producing the fruiting body. So Coltotrichum producing the asexual fruiting body is known as Acerulus. Dikaryotic spore of wheat stem rust. They are asking a dikaryotic spore. If you see all the spores, generally pycniospore, rhesiospore, iridospore, teleospore, all the spores are n plus n. So such kind of things. Among the all the spores, generally this dikaryotic spore means teleospore, which is a double-celled spore, right? So teleospore is a double-celled and also dikaryotic. And other spores are also dikaryotic, like teleospore is also, iridospore is also generally dikaryotic, but they're asking in the meaning of two cells, like contains two cells. And generally dikaryotic means n plus n. So best it is goes to teleospore. And the next one, which spore initiated the wheat stem rust in India? In India, wheat stem rust disease was initiated by iridospore. In foreign countries, it was initiated by asiospore. And but generally, in all the literature, it is mentioned that iridospore in India is the spore that initiate the wheat stem rust life cycle. The next one, blotter test commonly used to detect. So blotter tests are blotter pipes are generally used to detect seed-borne fungi. So seed-borne fungi only, not bacteria. Okay, seed-borne fungi. So nylon netting of tomato nursery is a practice to manage. So nylon netting is a practice that generally used in this tomato nurseries to manage a disease called tomato leaf curl disease. The last one, beans anthracnose is caused by 110. Beans anthracnose is caused by Colatotrichum lindimuthianum. Beans anthracnose is caused by Colatotrichum lindimuthianum. The function of Pycnia or spermatia in wheat stem rust life cycle was discovered by. So before there are 4 stages of wheat stem rust was discovered: stage 1, stage 2, stage 3, stage 4, all are Roman letters. But there was something was missing, the link that is a 0 stage. Zero stage is also known as a Picnial stage, also known as the spermatial stage, that was first discovered by Craig, C-R-A-I-G-A-E, Craig in 1927. And he also wrote a book, I think, Function of Pycnia of Stem Rust or something like that. He also wrote a book. And he's actually a Canadian plant pathologist, I believe. So the next one is spores of wheat stem rust is produced on generally five spore stages are there: pycnium, acium, iridium, telium, basidium, right? Pycniospores are produced on the upper surface, spores produced on the lower surface of secondary host. When we see the teleospores and iridospores, they are produced on the primary host, both are on the upper surface. I repeat, so primary host, that is the wheat, there are two spores we can observe, that is iridospores and teleospore, the two on the upper surface of wheat. If you see the secondary host barberry, on the upper surface we can see pycniospore, on the lower surface we can see acaiospore. So the answer is lower surface of barberry. Acaiospores of wheat stem is produced on barberry. If you want to mention extra information, that is the lower surface of barberry. The next one is first A virulent gene was isolated from which crop? First A virulent gene was isolated from a crop called soybean. From soybean, the first A virulent gene, that is to bacterial A virulent gene, was isolated around in 1954. The next one is first A virulent gene isolated from the pathogen. What is the pathogen involved in the isolation of the first A virulent gene? That is a bacterial blight of soybean. I repeat, 113, first A virulent gene isolated from a crop, soybean. 114, the first A virulent gene isolated from which pathogen? That disease is bacterial blight of soybean. Bacterial blight of soybean, which is caused by Pseudomonas syringae pathover The next one is Coco solan suit is transmitted by mealybugs. The scientific name of the mealybugs is probably Pseudococcus nigalensis, I think. So banana bunch top and cutter disease of cardamom is transmitted by, at least if you know one, the remaining the answer is same. Banana bunch top, I probably you guys know this. Banana bunch top is transmitted by black aphid. So along with this banana bunch top, one more disease, that is a cutter disease of cardamom, that is also transmitted by this black aphid, Pentalonia nigronervosa. Low pat test is used for, so low pat test is a test that is generally used for the identification of fluorescence Pseudomonas, or we can also call as Pseudomonas fluorescens. This L means leave and O means some kind of abbreviation will be there for this low fat. I actually forgot, I couldn't able to recall now. Leave and some kind of thing will be there, but they definitely use it to identify this fluorescent Pseudomonas or Pseudomonas fluorescens. The next one is the five-celled ascospore is produced by Meliola. Meliola is also called as Pestalozia. I think, I'm not sure. Probably no. That is different. Five-celled ascospore they are asking, not conidia. Okay. Five-celled ascospore is produced by Meliola. And five-celled conidia is produced. Whenever it comes to conidia, five-celled conidia is produced by Pestalozziopsis. 119 is Pestalozziopsis. Six-celled conidia is produced by Pestalozziopsis. So Pestalozziopsis and Pestalozziopsis. Okay. Five-celled ascospore is produced by Meliola. Five-celled conidia is produced by Pestalozziopsis. And six-celled conidia is produced by Pestalozziopsis. Potato golden cyst nematode remains viable in soil for how many years? So around 15 to 20 years it will be remains in soil, around 15 to 20 years. So citrus greening is transmitted by citrus greening is generally, if you see this citrus greening, generally half fruits are green and half fruits are ripened, so that's why it's called as citrus greening. So which is actually transmitted by probably psyllids, maybe. Yes, citrus greening is transmitted by psyllids only, Diaphorina citri, I think. The next one, citrus tristeza is transmitted by aphids, a species of aphid called Toxoptera citricida. And then 24, genome of Phytoplasma and Spiroplasma. So which means they're asking what is the genetic material of both Phytoplasma and Spiroplasma. So this Phytoplasma and Spiroplasma contains both DNA and RNA as a genetic material. The negative staining agent. So nigrosan is generally used as a negative staining agent in staining. Irvinia amylovora enters through Irvinia amylovora enters through nectaris, probably, or I think so. Trichomes or hairs, maybe, probably it's a nectaris. Please refer 126. I'm not sure. Probably it enters into the plant cells through nectaris. Then the next one is operon concept theory was lac operon concept. There is a concept called a lac operon concept. How this gene expression will work on. This is called a lac operon concept that was given by Jacob and Monads, two geneticists, I think so. So trap crop for Vorabanki. So linseed is generally used as a trap crop for Vorabanki. Of course, other trap crops also can be used, but generally this linseed can be recommended. So the indicator plant for tomato spotted wilt virus, they are not asking vector, they are asking indicator plant. Generally this tomato spotted wilt virus is transmitted by thrips, but they are asking indicator plant. Generally the indicator plant for this tomato spotted wilt virus is Nicotiana benthaminia. Then two ascospores are produced by there is a pathogen that produces only two ascospores, that is a phylactinia. Sex hormone in mucor. So this occurrence of this hormones was first reported and worked on by Anton de Barry and other scientists as well. This hormone that is produced by this mucor is trisporic acid A. Trisporic acid A is the sex hormone that is produced by the mucor. So melanin containing aprosoria can be observed in the fungal genera. It is there are plenty of genera can be there, like if you see Pyricularia produces melanized aprosoria, Coltotrichum also produces Cochiliobolus, Alternaria, verticillium, various kinds of pathogen are there. According to the options we need to go through, but generally the most importantly this melanized containing aprosoria means we can say this rice blast pathogen, that is Pyricularia varizae, or we can also call as Magnoporte grigia. Generally this pathogen, this aprosoria contains high concentration of glycerin as well as this melanin. So this melanin helps to create a huge turgor pressure that makes the hole into the epidermal cells of the leaf. So that is how it will actually. So if the more amount of melanin will be there, that creates a turgor pressure. So generally it can be observed in Pyricularia. If we can, if there is no melanin or this substance in aprosoria, that will be difficult for a pathogen. Generally we can say it cannot cause the infection because of this melanin, without melanin. So the sex hormone produced by the rhizophus. What is the sex hormone that is produced by the rhizophus? Before we have seen sex hormone produced by the mucor is trisporic acid A, but here this rhizophus can produce trisporic acid B and C. The next one is the inclusion bodies staining agent. Azurea generally is a staining agent used for inclusion bodies. I think we have in the general plant pathology we have mentioned different kinds of staining agents and all those things. Generally this inclusion body staining agent is Azurea. And the next one is the enzyme produced by the soft rot pathogens. Generally, the soft rotting causing pathogens produced in enzymes called pectinolytic enzymes. These pectinolytic enzymes are the tissue disintegration enzymes for the disintegration of tissues. We can take an example of soft rot of carrot. That can produce pectinolytic enzymes. You can see Pythium and Phytophthora that can produce some kind of cellulytic enzymes. So all are tissue disintegration enzymes only. So generally these pectinolytic enzymes are largely considered as, not largely, generally considered as this tissue degrading enzymes, of course cellulotic enzymes as well. And monograph of apple scab was written by first of all we need to know what is the pathogen of apple scab. Venturi inaequalis, right? Venturi inaequalis causes apple scab. The monograph of apple scab was written by William McCarty, McCarty. And monograph of wheat stem rust in India was written by. Monograph of wheat stem rust they are asking. So generally E.J. Butler wrote this monograph on wheat stem rust, general accounts on this wheat stem rust. And the monograph of a bead curly top was written by C.W. Bennett. Then 139, papillate stalked single celled teliospore is produced by. So for confusing us, they will tell 100 things. Papillate stalked, that's all not necessary for us. whether it is a single celled uromyces, double celled teleospore, like different types of teleospore we have already given in the section of fungi and as well as other sections as well. So there are some types of teleospores are single celled, double celled, multi-celled, parachute like teleospore, Ravenelia, multi-celled teleospore, something like that it will be there. Crust like teleospore, kidney like teleospore, or bean like teleospore, emilia, something. So such kind of things will be there. So just to see whether it is a single celled uromyces, double celled puccinia, parachute like Ravenelia, crust like, so such kind of things will be there. So to keep this single celled or double celled, we can answer this. So since they asked the single celled, so answer is uromyces. The next one is PR proteins are accumulated in. So they're asking where these PR proteins are accumulated. If you see this host pathogen interaction, generally it triggers hypersensitive response, and then followed by SAR and systemic acquired resistance ISR, and ultimately triggers this production of PR proteins, phytoalexins and PR proteins. So these PR proteins are generally there are certain kinds of PR proteins are generally identified so far. This PR proteins accumulates in apoplast and vacuoles. They accumulates in apoplast and vacuoles. 141 spermatial stage or sexual stage or zero stage of wheat stem rust fungus. So in the previous slides only we have discussed this zero stage is also known as spermatial stage, also known as sexual stage, also known as a zero stage, which was discovered by Craig. So this zero stage is known as a pycnial stage or pycnium. So the next one is the coconut basal stem rot. 142 coconut basal stem rot is otherwise known as, or also known as, generally this coconut basal stem rot is known as Tanjore wilt, because the disease was first identified in Tanjore by E.J. Butler, maybe probably, I'm not sure. Discovered in Tanjore, that's why it's called as Tanjore wilt. And before that also it was discovered in some parts of Karnataka, but it will become epidemic in Tanjore district of Tanjavur district of Tamil Nadu, and also rice blast also became epidemic in Tanjavur district of Tamil Nadu in 1985 probably. So the next one is coconut root wilt. Coconut root wilt is also known as Kerala wilt because it was first identified in Kerala. That's why it's called as Kerala wilt. Pepper yellows mottle virus is transmitted by this pepper yellows mottle virus is transmitted by mealybug. So mealybug is associated with the transfer of this pepper yellow mottle virus. Inclusion bodies of a cucumber mosaic virus. So generally there are each and every virus produces their own inclusion bodies. For example, if you see tobacco mosaic virus, X kind of inclusion bodies, such kind of things will be there. Generally this cucumber mosaic virus produces virus aggregate kind, virus aggregate, the name of this inclusion body is virus aggregate. The next one is the napiform, that is a turnip shaped teleospore is actually produced by Hemilia. So turnip shaped teleospore or kidney shaped or bean shaped iridospore, both, I repeat, turnip shaped teleospore, kidney shaped or bean shaped iridospore, both are produced by the pathogen Hemilia, which causes coffee rust, Hemilia vastrostix, right? So this napiform, this turnip shape is also known as a napiform, that is also we need to consider. The next one, the shape of teleospores of a coffee rust pathogen. So just now we told, right, turnip shaped, kidney shaped, and other names also will be there, like turnip shaped or kidney shaped or bean shaped or hump beaked. So generally these are all names. For 147 it is turnip shaped or kidney shaped or turnip shaped or kidney shaped or hump beaked. Such kind of names will be there. So 148 red leaf of oats is caused by, red leaf of oats is caused by a virus which is a barley yellow dwarf virus. 149 largest inclusion bodies are produced by, so PVX, potato virus X, will produce the largest inclusion bodies. The next one 150 inclusion bodies produced by alpha alpha mosaic virus. So this alpha alpha mosaic virus produces a crystalline type of inclusion bodies. The genome of furroviruses. Furroviruses genome is composed of single stranded DNA, RNA, single stranded RNA. Furroviruses genome is single stranded RNA. For example, I will tell you one more pathogen, Gemini viruses. So all Gemini viruses, their genome is composed of single stranded DNA. The next one is 152. Potato wart is an endemicon. Potato wart is endemicon. Where they are asking? Generally, it is endemic on Darjeeling district of West Bengal. It is a district where potato generally grows more. In this area, this Potato wart is an endemic disease. The next one is golden cyst nematode of potato is endemic on. Golden cyst nematode means Nilagiri. So it is endemic on Nilagiri. I repeat, potato wart is endemic on Darjeeling. Golden cyst nematode is endemic on Nilagiri. This both pathogen having the quarantine, domestic quarantine importance, where they cannot spread the infected material from the infected area to or infected state to other states for the propagation purposes. I repeat, potato wart is endemic in Darjeeling, whereas golden cyst nematode is endemic on Nilgiri Hills. These two are the domestic pathogens, domestic quarantine importance. So 154 biocontrol agent for Guava fusarium wilt. So Gava or Guava fusarium wilt. It is a little bit I would say generally the answer is Aspergillus niger. Generally Aspergillus niger causes some kind of moldy growth or some kind of infections on various kinds of post-harvest fruits. But generally there is this Aspergillus niger can be exploited against the management of Guava fusarium wilt. The next one is toxin theory was developed by who developed the toxin theory, they are asking. So Gauman, so E.A. Gauman was the first one who developed this toxin theory. So the next one, which genera of downy mildew they germinate through germ tube, they are asking. They germinate directly through germ tube. So the genera is Peronospora. Peronospora generally directly germinate through from this germ tube. So maybe other pathogens also might be there, but I can remember this Peronospora for right now. And common name of Ziphinema, so dagger nematode, Ziphinema index, dagger nematode, right? So the next one is citrus greening is also known as yellow dragon disease, also known as Huan Long Ping. So it was first identified in China, China provenance around 1920s or maybe probably 1930s, and it is believed that in this citrus greening citrus greening specimens was collected from India in around 1830s and 1840s itself, but officially it was reported from China. The citrus greening is also known as Huan Long Ping or yellow dragon disease. Black tip of mango was reported by, they are not asking caused by, first reported by. So black tip of mango first reported by Das Gupta. Das Gupta was the first person who reported this black tip of mango from Bihar. So the next question is black tip of mango was first reported in, okay, again same Bihar. It was first reported in Bihar. The next one is 161. Cigar and cigar shaped inclusion bodies. They are asking cigar shaped inclusion bodies are produced by. So cactus virus X generally produces this cigar shaped inclusion bodies. I repeat, cactus virus X produces cigar shaped inclusion bodies. So primary spread of apple scab pathogen. So primary spread is generally this apple scab pathogen overwinter survive in the apple leaves. So that was a fallen leaves in the form of pseudothecia. This pseudothecia can serves as a primary spread. The spores can survive in the infected leaves, that is that too on fallen leaves in the form of pseudothecia, where the pseudothecia is actually formed on the leaves, and this will actually serves as a primary spread. The next one is slow decline. Of course, anyway, there are three kinds of declines are there that was given here: slow decline, spreading decline, quick decline. I think slow decline is generally caused by nematode. I repeat, slow decline is caused by nematode. The nematome is Tylenchulus semipenetrans. Spreading decline is caused by banana burrowing nematode, Rhadophilus similis. So slow decline and spreading decline are both are caused by nematodes. Slow decline is caused by citrus nematode, that is Tylenchulus semipenetrans, whereas a burrowing nematode Rhadophilus similis, banana burrowing nematode Rhadophilus similis, that causes spreading decline. And quick decline is generally caused by virus, that is a citrus tristeza virus, which causes us quick decline. Three kinds of declines are there: slow and spreading declines are caused by nematodes. Quick decline is caused by virus, that is a citrus tristeza virus. The next one is root tip curl is caused by the infection of root tip curl is caused by the infection of Ziphinema. It is also nematode. So Ziphinema is responsible for the root curling tip. The term toxin was first used by, I think, Ludwig. I'm not sure. Probably the term toxin was first used by Ludwig. But the term vivotoxin was given by Diamond and Wagner in 1953, and the concept was given by probably T.S. Sadasivan. But the term toxin, general toxin, was given by probably Ludwig. I'm not sure. Please refer this 167. And 168, which plant hormone act as a pollutant disease? So there are many plant just a minute, plant hormone they are asking, not plant pollutants. So generally this ethylene act as a plant pollutant. If you see this plant pollutant that can cause plant diseases, ozone, peroxycyl nitrate, ethylene, some kind of will be there. Among all this pollutants, ethylene is also a plant growth hormone, right? So this ethylene is generally causes plant pollutant. Sometimes it can cause a disease as well. The next one, wound tumor hormone is transmitted by the vector. Wound tumor hormone, they ask. Wound tumor virus, probably it's not hormone, okay. Wound tumor virus, it is also the biggest virus, 70 nanometer probably size in terms of, probably it is transmitted by leafhopper, Agalia constrictor. The next one is resting spore of Plasmodioporomicetes, resting spore. So the resting is generally cystic when produce. Uh, Juice spores, generally resting is a cyst only. Again from the cyst again juice spores will form, right? The experimental host of Phytoplasma. For Phytoplasma, the best experimental host is periwinkle, Catharanthus roseus. And other seeds also, other plants also might be used like rapeseed, cauliflower, something will be there. But generally periwinkle is the best one for this Phytoplasma. Pinwheel inclusion bodies are produced by Poti virus. And leaf drop streak symptom is produced by, again same, potivirus. So both this pinwheel inclusion bodies and the leaf drop streak symptoms both are produced by potivirus only. Progametangema of Zygomycota is called as a suspensor. Suspensor is the general term that is used for this progametangema. So if you see this zygospore, either side of the zygospore it holds a holding kind of structure is known as a suspensor. The progametangema in Zygomycota is known as a suspensor. 175, which mushroom is called as a mycoremediation or mycoremediation of pollutants? There is a mushroom called Pleurotus, oyster mushroom, Pleurotus ozarka as we used to call, right? So this Pleurotus is the best having this mycoremediation properties, so it can use to to take all the pollutants out. And actively discharged spore is, which spore is generally considered as actively discharged spore? Ballistospore is actively discharged spore. Statesmospore is a passively discharged spore. So 176 is actively discharged ballistospore. Again, okay. Passively discharged spore again came here. Statesmospore. The next one is genome of molecules. Molecules means Phytoplasma and Spiroplasma both are combinedly called as molecules. Their genome is composed of both the DNA and RNA. In the previous slide only we have discussed, right? So genome again same, it contains both the DNA and RNA as a genetic material. Phytoplasma, Spiroplasma are collectively called as molecules. Now it's called not tentacles, molecules only. And 179, alternation of generations are found in, just changing these generations are found, best known is found in Allomyces. This is the genera which is created by E.J. Butler, father of Indian plant pathology. We can also call as father of Indian mycology as well. So this alternation kind of generations are found in Allomyces. Vomit toxin, which of the following is a vomit toxin? Dawn toxin, there is a toxin called dawn toxin. Deoxynivalenol is generally called as vomit toxin. Deoxynivalenol. Cru disease of humans is generally caused by, there is a pathogen called prions. We can't call as a pathogen, it is an infectious protein. So prions are the malfunctioned infectious protein which causes a disease in humans called Cru disease. In animals it causes a disease called bovine spongiform encephalopathy in animals, again due to the same prions only. It was first discovered by Stanley Prusiner, for that he got Nobel Prize in 1994 probably. So Cru disease in humans is caused by prions. Bovine spongiform encephalopathy is due to again prions only. What is prion? Just now I told you it is a malfunctioned protein or a diseased protein or infectious protein we can say. So phloem necrosis of coffee is caused by generally a pathogen called Phytomonas species, probably Phytomonas leptovasarum. And frog spot of apple is caused by 185. Frog spot of apple is caused by Botrosparia obtusa. Fungal transmitting viruses are known as furroviruses. fungal transmitting viruses are known as furroviruses. For example, genus like Syncytium, Wolfedium, these are all the best genus of fungi that can transmit the viral diseases. So bendy yellowwing mosaic virus was first reported in India by Kulkarni was first reported this bendy yellowwing mosaic virus. The term slow viral diseases are related to, it is generally the slow viruses are related to prions. So slow virus because it can kill the host very slowly. Mini viruses, that is the term related to viroids, because the viroids are 100 times smaller than that of viroids. That's why it's known as mini viruses. The term mini viruses are related to viroids, slow viruses are related to prions. Sporulation can be quantified by, so what is the equipment that is used for the quantification of this spores? Hemocytometer. There is equipment called hemocytometer which is used to quantify the spores. Lala France disease or X disease or water stripe disease, probably this is the first disease that occurs to mushrooms. So which is caused by, phorid flies. Phorid flies are generally involved in this Lala France disease. And the next one is Rhizophus stolonifera. Rhizophus nigricans produces an acid called fumaric acid. Fumaric acid is the acid which is produced by Rhizophus stolonifera. Sclerosum producing an acid. Again same, Sclerosum also produces an acid called fumaric acid only. So Rhizophus circinens is produces an acid called lactic acid. They are asking this byproducts. Normal size of microscopic slide. So microscopic slides are size ranges from 76 to 26. So this is the common size of microscopic slides. Normal size of cover slip they are asking. So cover slip is generally 25 to 25, or maybe probably 18 into 18. Aplanetism can be observed in. Aplanetism means only one kind of juice spores in the life cycle. So that is generally observed in Zeolina. So biplanetism or this kind of mechanism, biplanetism generally can be observed in Saprolegnia, where it can produce two kinds of juice spores, right? Motile spores. Aplanetism can be observed in Zeolina. Monoplanetism can be observed. In monoplanetism, we can say Pythium Phytophthora monoplanetism. Diplanetism, again just now I told you, Saprolegnia is the best example for diplanetism. So polyplanetism means Acaila. So Acaila is the best known for polyplanetism. So this is Saprolegnia, diplanetism fungi, and polyplanetism, Acaila fungi. Both, even though they comes under Oomycota group of fungi, their cell wall is made up of chitin. Generally, Oomycota group of cell wall is made up of cellulose. But even though they comes under this Oomycota group of fungi, their cell wall is made up of chitin. There are a couple of fungi like Acaila, Aphodogyla, Saprolegnia, and Leptometas. They are all the fungi. They comes under Oomycota group, but still their cell wall is made up of cellulose. For further more information, students can refer this book, A Vision into Plant Pathology, a complete student version. Further doubts and clarification, student can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.